Hey, grade 11s. I've decided to do this in two shorter lessons just so that you guys are maybe not pushing too much knowledge down your throats. So we're going to take this not easy, but we're just going to split it up into two lessons. This shouldn't be too long, so let's do it quickly. All right, so we're now going to be looking at the biodiversity of animals. So I want you to keep all that information of phylogeny in your head that we did earlier, and I want you to be able to apply it to animals as well. So I'm going to teach you all the characteristics, much like I did in plants, and then you guys are going to have to draw up a phylogeny tree for animals as well, but for only 6 different phyla, nothing more than that. All right, so and like if you want to do more, we're more than welcome to when we get back to school, but for IAB we only need to do these 6. Okay, please note that all of these are multicellular organisms. Okay, so they've been through the process of some sort of differentiation, and there's more than one, and they all slightly do different jobs. They are all eukaryotic. All right, none of them are prokaryotic, so none of them are bacteria. And finally, I just need to talk. I might use the word simple, and I might use the word complex sometimes. Come, Knox, come say hi. Say hi, Knox. It's my puppy, Knox. We need to talk about simple and complex because to a large extent, when I talk about simple, I'm just talking about slightly less evolved individuals versus slightly more complex or higher evolutionary evolved. So it's not to say that the simple individuals aren't complicated, but it's just to say that they maybe aren't as well developed, okay? But they still function extremely well. All right, these are the 6 phyla we're going to be talking about. We're going to be talking about the Porifera in the bottom right-hand corner. Then we'll be doing the Cnidaria, which is in the bottom middle. Then we'll be talking about the Platyhelminthes, the Annelids, the Arthropods, and the Chordates. You as a human are there in the Chordates. The general word for Porifera is sponges. Cnidaria are the sea anemones as well as jellyfish. Annelids are earthworms. Platyhelminthes are sea slugs. And arthropods are all insects, whether they are 6-legged, 8-legged, or 10-legged. Yes, we get 10-legged individuals. All right, so please won't you draw this for me? I'm always going to ask you questions from this, and I give something very similar to this in your test, or I might just use a chunk off of it. So right at the bottom left-hand corner, it has a single-celled ancestor. Then we had Porifera, Cnidaria, Platyhelminthes, nematodes, mollusks, annelids, arthropods, echinoderms, and chordates. I want you to cross out when you do this. Don't cross it out as in like it doesn't exist, but we are not going to learn about nematodes, mollusks, which are all of your, what do you call those things again? Clams and snails and slugs and octopus and cuttlefish. And then like cross-eyed echinodermata as well, because those are your starfish, and we're not going to talk about them. So what you can see at the bottom, we have multicellular, so following that line to the right-hand side. Then we have tissues, which means like tissues start to develop. So like skin cells, muscle cells, blood vessels, that kind of stuff. Then we have 3 germ layers and bilateral symmetry. Then we have protosome development. So when you are a ball of cells, remember we spoke about in grade 10, and then like when fertilization occurred, then they had to rapidly multiply to create the individual that you are today. Like in old times, especially in humans, there's like a dip that forms, and it almost creates like you've pushed your finger in some dough. And when that happens, that actually becomes that keeps on growing and creates the mouth of the individual. Okay. Then we have the pseudocoelom. We'll get to that later. We have a coelom. Then we have the deuterostomes. Now this is where things get crazy because this is what humans are, and that little like piece I was telling you. So in other like animals prior to that, it becomes the mouth. That hole that forms that grows all the way through that ball of cells actually becomes is the anus, and then it becomes the mouth. So you literally like inverted yourself, and a tube grew through that entire structure. There we have radial symmetry again, as well as in the cnidarians, and then yeah. So this is just a lovely version of a cladogram. So this is flagellate on the far left hand side, which is like an ancient ancestor. These were just one type of cell. Then we had porifera, cnidaria. Then we go into like a whole bunch of crazy bits and pieces, and I don't need you guys to know all of them. So the ctenophores are actually a group of extremely poisonous jellyfish. Then we have nematodes. Some of them can actually cause real damage in humans. I don't know if you've ever heard of elephantitis. Go and Google it. With the mollusca, annelids, there's little cnidarias, rotifera, nematoda as well. Sorry, that's nemertea on the far left-hand side. And then so on and so forth. But we'll get to all those little bits and pieces a little bit later. Again, you guys can pause this if you want. You don't have to draw this. You have to draw the previous one. Right, same, exactly the same, just in a slightly more complex version. I'm sorry the words are a bit small, but to try and squeeze the whole thing in was a bit difficult. So again, you can see at the bottom there is porifera, cnidaria, ctenophora, platyhelminthe, nematera, nematoda, rotifera, mollusca, annelida, arthropod, echinoderms, and chordates. So you can see that chordates are the least related to porifera. Chordates are actually closely related to echinoderms. So we are more closely related to starfish than any of those other individuals over there. And you can see kind of what split off at each time. So right at the top over there, we have deuterostome, and then underneath that, a protostome. And then we have segmentation, and that's important for those arthropods and annelids. Notice that a snail does not have segmented body parts, whereas an insect and a earthworm does. Each one of those rings is a separate segment. And depending where you cut the earthworm itself, if you cut it correctly, it can regrow an entire new earthworm from that. All right, so these are the key body features that we're going to be looking at. We're going to be doing body symmetry, cephalisation, the number of tissue layers developed from the embryo, development of a coelom and a blood system, and the number of gut openings. So symmetry is important because you notice if you cut yourself down the center of yourself, you will be able to create two identical halves. Sure, you're not going to be completely symmetrical, but you're going to be quite close to symmetrical. Then we have radial symmetry, which is the idea that if I was a circle, you could cut me along any one of the axes around and I'd create a perfectly mirrored image as long as you cut me along a diameter. Then we have your asymmetrical versions, and I'll show you lots of pictures of those, but if you could pick from the previous slide, that was your sponges. They lack symmetry. And we'll find out why symmetry is important a little bit later. And think of why it is important that you have 2 eyes and 2 ears. And like your ears are on either side of the head, and the nostrils are either side of the body but close to the center. Why are your arms so far apart on the opposite side? We'll talk about that later. Then we're going to do cephalisation, which is the creation pretty much of a brain or a head. So cephala, meaning of the head. Okay, so have you ever heard of encephalitis? That is swelling of the brain, and it's a really dangerous disease. So you can go ask your parents about that. A lot of the time, some people suffer quite badly with encephalization. Then we have the number of tissue layers which develop from the embryo. You'll notice that some have 2 layers, some have 3 layers, and some have 3 layers with a hole in the middle. Then we have the development of a coelom and a blood system. Now remember, when I spoke to you about plants, I said to you that vascular tissues were super important in the sense that it allowed those plants to grow bigger and taller because they had a way of actually pushing around all of their nutrients so that it could actually move to the tips of the branches, which allowed it to grow further. And it's the same with animals. A blood system allows you to move nutrients as well as water and oxygen and a whole bunch of stuff, which allows the individual cell to grow bigger and stronger. Eventually, things as big and large as whales, then you have to have the addition of water where there's extremely high pressure. Then the number of gut openings. So you have 2 gut openings. You have your mouth and your anus, whereas you have your sponges or porifera, they only have 1 gut opening. Then you have something like a jellyfish, which also only has 1 gut opening, so its mouth is its anus, where you have 2. So count yourself as lucky. All right, so let's look at the I'm going to explain each one of those, and then I'm going to expose you to each one of the 6 independent phyla, just so that you have an understanding before. So write these down, make sure that you understand it. I've also thrown in some examples over there. So asymmetry, they have no symmetry whatsoever. I've put a little diagram on the right hand side. So the left one is symmetric and the right one is asymmetric. So A meaning the opposite of symmetry, so it is not symmetrical. They cannot be divided into two halves that are mirror images. Example, sponges. Then we have radial symmetry, super cool, arranged around a central axis. Okay, the central axis is in the center of that starfish. Can be cut in more than one vertical plane through its center to obtain mirror image. So remember, one doing that is, well, going up to the ceiling from the floor is vertical, going from wall to wall from left to right is your horizontal. So there's no left and right side. There are multiple bits and pieces to it. They usually have a mouth at one end, example jellyfish, and in this instance starfish. Then we have the bilateral symmetry, which is what you are. So the body parts are arranged on either side of a central axis, so left and right, and can be cut into two mirror images in only one plane, so left and right hand side. The dorsal and ventral side, as well as anterior and posterior end. Now, in case you forgot what that is, I've put a lovely little diagram of a horse in there, and you'll be able to remember what dorsal and ventral is. So your dorsal is your back, and your ventral is your stomach. Your posterior is from your bum backwards, and your anterior is coming from the top of your skull all the way up. It's suited to animals that move about because it gives you a sense of direction. You now know what front is, what back is, what left is, what right is, and you can maneuver based on your senses to be able to tell you what is going on. Sorry, ladies, the video is going to jump up. Like, went through too many slides at once quickly. All right, so here's just some examples of different kinds of symmetry. We have radial symmetry at the top over there. The top left-hand corner thing is called a sea anemone. I will also be teaching you about hydra. No, not hail hydra. Then on the right-hand side is a pot. And you can see from that pot, you can cut it along the diameter as long as it is in a central axis. So central axis is a point that is directly in the middle of the object. Then we have the lobster at the bottom left-hand corner over there. And we have a spade, which is also bilateral symmetry. And the top are radial symmetry. All right, almost finished now. Cephalisation. A characteristic of bilaterally symmetrical organisms. So only once you are bilateral can you have a cephalisation or the formation of a head. Remember, as a radial individual, you have multiple parts. So your head would technically be in the center, but there's no actual development of the head. And what is cephalisation? It's the concentration of organs near the anterior end of the organism. All right, so you can see on the right hand side over there, it says the nervous system of a flatworm. Those are the most cute things ever. I will hopefully send you, get you some pictures in class so you guys can have a look. I forgot to include one in here, but it's actually called a planaria. Google it. It is really cute. And I know you're gonna think I'm weird, but it really is cute. And you can see that it has a nerve cord, and then right at the top there it has something called the anterior cerebral ganglion. Now, the definition for a ganglion is a group of nerves that have collected together that act as a waiting point for nerves or as a primitive brain. All right. In humans, however, our cephalisation or formation of a head has led to us having that beautiful brain with ears, nose, tongue, lips, skin, the works. All of your senses are over there. So that's where all your sense organs are. It's for has your feeding appendages as well, because remember, you don't necessarily want to put in something in your body without getting it to your senses first. So like when you walk up and you go and buy an apple, let's say you were buying an apple and a whole bunch of apples in front of you, you would look at it and if they all look the same, then you would pick it up and feel it. After you felt it, you will then smell it. If it smells okay, then you're going to eat it. So, putting that feeding appendage really close to your nervous system or sense organs really helps when you're eating. Finally, it creates nervous tissue. So over a time, that nerve cord or those nerve tissues start to develop and become extremely highly developed. and then we create a control center while using these nerves as they're able to communicate between each other. Also, just as a side note, please also remember that when you are walking into any establishment, you are often going to walk in with your head first, and that is because you have all of your senses. So a little bit later on, when you're walking around the house, I want you to like go and try and call your mom. And when you pop your head into the kitchen, what is the thing you do? You put your head around. And when you put your head around, it's actually so that you can see if there's any like danger coming your way, or it tells you quickly, oh, mom is here, or mom is not, or dad is here, or mom is not here, or gran, or auntie, or puppy, or whatever on earth is you're looking for. So the concentration of tissue forms simple brain in primitive animals and complex brain in highly advanced animals. Doesn't sound like I have a very complex brain at this point. Animals move freely. They enter environment head first. All right, and that's why often a lot of like very weak herbivores they pop their head out first as well to try and pick up if there's any dangers, and then they go running about. So they're able to take food, danger, and other stimuli and respond super quickly. Okay, so that's why this idea of having a head is so important, and especially in large mammals like us. All right. Okay, I'm going to stop it for there now, ladies. Next time, I'll get to the tissue layers, blood systems, coelom, and gut openings. And then after that, we'll do the different phyla. All right. I hope you have a great day, ladies. See you soon. Bye.